Well, good evening. So that's me. I'm a forest pathologist. I work for the Department of Natural Resources. I've been there for a while now. And there's a picture of me over to the left there with my most trusted tool, the Pulaski. If any of you are not familiar, that's a tool that is very common in wildland firefighting. And I found it to be a really useful tool for a forest pathologist. It has both a digging side and it has a cutting side. So it's really, I'm looking at an upturned root system there. Even if I need some help looking underneath the bark, looking at bark beetles, it helps me get underneath the bark there. So anyway, it's one of the tools of the trade for forest pathologists. And so I guess I have about an hour or so. I won't keep it too late tonight. And just give you sort of a survey of some of the common tree diseases that you're likely to see in your forests here in Western Washington. And why are we concerned with diseases of forest trees? Well, one reason we're concerned is because of their ubiquity. These diseases are everywhere. Wherever there is a tree, there's a pathogen associated with it. And this is a map of the distribution of just one disease. And you can see its distribution, that little teardrop, that's, you all recognize this as the Palouse country, so there's a lot of grass. But even grasses have diseases, but we're going to save those for another speaker. Diseases are prevalent throughout the forest, and they're problematic as well. And here's, this is a graph of numbers that were projected based on estimations and models. But it's over a 15-year period, and what they tried to figure out is how problematic are these various insects and diseases. And so on the y-axis we have mortality measured in board feet. And over this 15-year period, root diseases are projected to cost the economy over $1 billion board feet of mortality. We're nearing the end of that period, so it'll be interesting to see once we get down the backside how close those predictions were to reality. But regardless, they're recognized as significant agents of mortality. So the prevalence and they're problematic. So there are lots of agents we saw from the previous slide. There are beetles and there are root diseases. We sort of generically refer to these as disturbance agents. In the old days, we used to call them pests. But we recognize over time that there's a lot of value that these pests, depending on the context, can provide to forest ecosystems. And so there are several of these agents of disturbance. I think one of the reasons why they got that term disturbance agents, because there is a paradigm in many foresters' minds that in order to get from a young stand to an old growth, you go in a straight line. And if you've been around long enough, you realize that all along the way, the plagues, for example, that might influence the trajectory of that particular stand. If you've been around here, you've experienced windstorms, right? That out on the coast, wind blows 110 miles an hour and just blow down acres and acres of timber. Wildfires, and so foliar diseases, you know, they disturb that linear trajectory from young stand to old growth. And some of you were here in January, I presume, to hear my colleague Glenn talk about insects. And so the other group that we're going to talk about tonight are fungi. There are other organisms that cause disease in conifers and other forest trees, but none more so than fungi. And fungi, they belong to a really large kingdom of organisms, right? And so here's a slide that just reflects some of the various roles that fungi play in a forest community, right? Anywhere from saprophytic roles, so they're the decayers and the decomposers, and they're the recyclers of nutrients. There are some fungi that are mycorrhizal. They form symbiotic or mutually beneficial relationships with the roots of trees, such that there are many trees that couldn't grow in the forest in the absence of these mycorrhizal fungi, okay? Lichens are a symbiotic relationship between a fungus and an algae, right? Here's a little critter chewing on a mushroom. And chanterelles and other edibles, we're familiar with those. So we're gonna narrow our talk down this evening to just look at those pathogens, the small group, relatively small group of pathogenic fungi. And pathogenic fungi are generally tree killers, and they can kill single trees, one here and one there, or they can kill large patches or pockets of trees several hectares in size. And they're fungi, so let me just sort of, if you're not familiar, reintroduce you to the structure of a fungus. Really unsophisticated, just about as basic as you can get. There are generally two parts to the fungus. Many of us are familiar with the reproductive part, the toadstool or the mushroom. Conchs, those woody perennial tissues, structures on the side of trees are variations on this mushroom, which are basically the reproductive or the spore-producing structure of the fungus. And depending on the type of fungus, they can be really ephemeral, really short-lived. They exist for a day, a few hours, sometimes for a couple of weeks. Sometimes, in the case of these woody conchs, maybe for a couple of years. Their primary function is to create spores, disseminate those spores on the landscape, and once that job is completed, the function of that structure is also completed. But what persists for generations is this, the vegetative portion, the single strand hyphae, the physical body of the fungus, collectively referred to as mycelium. And you can see this web-like structure of tissue. It's those threads, those strands that are coursing their way into and around the cells in a Douglas fir needle, in the case of Swiss needlecast. They're the threads that are working through the bolt of a Douglas fir in the case of heart rot, and they're the threads that are coursing through the soil looking for sugars in the case of root diseases. And so here we have an example of a root rotting fungus. And so what we have here is we just took a cross section through the soil. Okay, so we removed half the soil so you could see what's going on underground. And there's our fruiting body. In this case, it's of a armillaria fungus, and we'll talk about that in a little bit. But maybe a little hard to see are these black threads. Okay, and these are the vegetative portion of the fungus, and they are heterotrophic. So when it comes to their mode of nutrition, they're a lot more like humans than they are like trees or plants, which are autotrophic. Autotrophs, when they need sugar, They simply expose their leaves or needles to sunlight, and through photosynthesis, sugars are created. Fungi, like us, we don't have that ability. We need to go hunt, we need to go gather, we need to go to the store, we need to go to the kitchen, and these threads are looking, just like us, they're looking for sugar. And so they're coursing through the soil, in the case of a root disease, looking for these sugar-rich areas in the soil, which are generally associated with trees, right? Because trees are leaking sugars all the time. The sugars are manufactured up in the crown, they're translocated into the roots, and we can see these roots, they're being abraded by rocks in the soil, maybe they're being nibbled on by small mammals under the ground, maybe the wind is blowing and the roots, the anchors are snapping, just enough to crack open the cell wall, and they're leaking sugars. And so these threads are sensitive to those sugars. And you can see what's happened is these threads have enveloped a root. Now, of course, they're different from humans in that we have our stomach on the inside of our body. We ingest food. These threads excrete enzymes. So out of these threads are coming caustic enzymes, and the roots are being bathed in these digestive enzymes, and so the cell walls are being exposed to this digestive enzymatic tissue that breaks open the cell walls. Okay, and as the cell walls are broken, the contents of those cells are liberated, and they leak sugar, and that's what these hyphal threads are eating. Okay. That makes sense to everybody? So what does a tree look like? Those roots are being rotted, right? These roots, they're important for anchorage, they're important for movement of water and nutrients, and so obvious symptoms are rot. Trees whose roots have lost their function, their ability to absorb nutrients and waters, you see a significant reduction in growth. By that I mean a lot of times these conifers, Douglas fir and others, they develop a flat top, right? They can no longer get those nutrients and water to the very top of the tree, and that limits height growth. And so you see a rounding on the top of these trees. Oftentimes you see, we refer to as a stress cone crop. It's not uncommon for young trees, even as young as 10, to produce cones, but year after year, there's oftentimes a stressor being imposed on those trees that are forcing them to, it's in a sense a last-ditch effort to leave progeny behind. Interestingly enough, many of these cones, the seeds within those cones are not viable. So it's sort of a lost cause. Another symptom would be chlorosis, and you can see that here, yellowing, in contrast to this tree immediately right next to it. Oftentimes you see the crowns are more transparent, right? They're not absorbing all the water and nutrients that they need, so they're unable to support the full canopy of foliage, because that costs a lot. And so you can see a lot more blue sky or transparency through that. And oftentimes on resinous species, Douglas fir is a resinous species, western red cedar is not, you'll often find this resinosis reaction from the tree to being injured. In this case, the injury is the result of infection. Oftentimes underneath the roots there. You probably, sometimes this looks a little bit like red turpentine beetle that maybe Glenn talked about a little bit. Red turpentine beetle is a bark beetle that preferentially infests the lower bowl. And so sometimes you can see in resinosis like this caused by a red turpentine beetle. And just as a reminder, these root rots, the fungi that cause these diseases primarily spread along the roots. And so that's demonstrated in this particular slide. So here's an old case-hardened, sun-bleached stump of a Douglas fir that was cut decades ago. And a young Douglas fir was planted in proximity to that stump. And everything was fine until the roots of this 8-year-old Douglas fir came in contact with the roots of this probably 30-year-old stump. And the fungus remained on that stump all that time and was just sort of waiting, if you will, for a victim to show up nearby. In this particular case, the fungus didn't move. This is a laminated root disease. The fungus merely remained on this old stump, and it wasn't until the roots of this tree grew in contact with that the fungus was able to make it to this young tree, get a breath, and then pursue its duty, really, to continue killing trees.
So laminated root rot doesn't move? It waits and lurks?
Yeah, interestingly enough, we've done studies where we've taken stumps like this, big stumps, 36-inch stumps. The tree is dead. The top gets cut out. We've excavated that stump and we've put little colored tacks, thumbtacks, on the margin of decay. Plunk. And then we've covered it up, and we've come back 5, 10, and 20 years later, and the fungus hasn't advanced beyond the margin of the tack. The fungus, Coniferporia sulfuracins, previously Polinus sulfuracins, and before that, Poria warei. Some of you old timers might recognize it as Poria. That's sort of the nature of how the fungus moves, but it's not a very strong competitor. Okay, it's a very effective pathogen. So its ability to overcome a susceptible host is largely resides in the fact that on a dying tree, if you will, there are not many other fungi. The tree is still alive, it's still producing resin, and it's effectively limiting other fungi from advancing into this weakening tree. And Coniferporia, the fungus that causes laminated root rot, does have a competitive advantage as a pathogen. Its ability to excrete these caustic enzymes, to absorb sugars. So it has an advantage over other fungi. And so that's how it gets a toehold and girdles the tree. But once that tree is killed, there's an array of other fungi that have been sort of hanging out, saprophytes, right? And they're better competitors at decomposing and decaying dead tissue than is the laminated root rot fungus. So we look at tree symptoms, and now we can look at stand symptoms. So, you got to keep in mind how that fungus sort of makes its way along root systems, right? And so we often develop these pockets of decay. And so here's a little schematic. And of course, the roots are underground. The red roots, of course, are infected, and the green roots are not infected. So you can sort of imagine this opening here is sort of the opening here where all of the trees, they're sort of laying on the ground, sort of a jackstraw area. And here's another image here taken from the center of the pocket. When you look outward, you can see some of these snags. Here's one, and here's one, and here's one. And then you get a little further out, and you can see there are no fines, a little further out. And then all the way at the edge here, you have, this is a thinning. You can see this is a tree that probably looks like this tree here, in the sense that the crown looks reasonably healthy, but the roots are infected, and so it doesn't have the same vigor as does this tree on the right or the one in the background on the left that likely is not yet infected. But these are very sort of classic stand symptoms. You know, you're just walking through the forest and all of a sudden you come to an opening. I always wonder, well, how did this get here? And oftentimes it's these root pathogens that are creating these gaps in the forest. Sometimes, especially if time has passed, you'll be walking through, say, a Douglas fir forest and all of a sudden come upon a stand of western red cedar. And I asked myself the same question, how did this get here? And likely western red cedar, the gap wasn't all that big, right? So it's still shady, maybe too shady for a Douglas fir to establish itself, but certainly not too shady for western hemlock or western red cedar. And so in time, that's how these pockets get colonized by sometimes resistant or tolerant species, generally more shade tolerant, if not also more disease tolerant. So just briefly, there's probably half a dozen root diseases that you're likely to encounter on a property of any significant size. But the three most common, as I mentioned earlier, there's laminated root disease. This is a chunk of decayed wood that we pulled out probably from the roots here, and you can see how we should call it delaminated root rot, because that's really what's happening to these woods along the annual growth rings, right? So there's early wood and there's late wood, and one decays a lot easier than does the late wood, and so that breaks down very quickly. And you can see different species. The fungus has preferences, just like you and me. You know, you like steak, I like fish, whatever. And so these different pathogens out there have preferences themselves. You know, they've evolved over eons with this very unique competitive advantage. And so it takes a lot of energy to specialize and be able to overcome the defenses of any single host. And so, and generally, that's what explains a lot of this the susceptibility across species. And so, laminated root rot, highly susceptible, Douglas fir and grand fir, and on the other extreme are hardwoods, which are immune.
How about pine?
Pine
Ponderosa or western white?
Yeah, I would say intermediate in terms of resistance. Probably, so you can see there's a continuum. We have highly susceptible. Basically, that means I'm just going to look at you and you're going to wilt. Right? and then there's tolerant. So that would be another category that's not represented here. And a tolerant is like, okay, you can have my leg. You can have my leg, you know, and I'll tolerate you. You're not going to get me unless maybe it's droughty, or maybe, you know, I'm stressed by bark beetles or something else. And only under those unique situations, am I then going to give up the ghost because you got my leg. And then there's resistant, which like, no, you know, whatever you've got, you may in fact get a toe. I'll give you a toe. But no matter how much you have of that toe, and no matter how tightly you hang on that toe, that's all you're going to get. And so a resistant group like a western red cedar, we never see mortality caused by this disease. So there's a continuum.
But the positive,
Is it hemlock in there too?
Hemlock, so the two hemlocks that we consider are mountain hemlock, which is highly susceptible. Mountain hemlock would be in this category, whereas a western hemlock would be in the tolerant category. And the pine would also be in the tolerant category.
Yeah. The big toe and the leg, is that scientific?
Yes, look it up.
It's a good
Seems to
idea.
work.
If it works, don't complain.
Fine.
So another one that's pretty pretty common. It's common in the sense that we refer to armillaria root disease. Armillaria, you're talking about armillaria, but it's a bit of a misnomer because armillaria represents the genus. And within the genus Armillaria, there are probably a dozen species of Armillaria. A couple of them, Astoes and Solidipes, are highly pathogenic. Others, Melia and Gallica, pathogenic on hardwoods but not on conifers. So we often, and they're also, Armillaria in general, are very good saprophytes. If you were to put a pin on the Armillaria on a dead tree, in 6 months it would just breeze right by that pin stalk, and it would move up the stump, and it would move out the root system, and it moves everywhere. And the other unique feature about Armillaria, this is the primary diagnostic, is this underneath the bark. You can see we moved the bark of this tree. We have this is the mycelia. It's an aggregation of these hyphal strands. And it's very distinct. You can sort of imagine it coming up through the root system and girdling the tree. That's sort of what I think of when I see this. It advances like a fan does. And you can see it's very thick. It's like latex. You can pull it apart, and it's very, you don't need a microscope for this one. Occasionally in the fall, for maybe a week, maybe 2 weeks, you'll see these honey-colored mushrooms. In the case of Armillaria. Oops, didn't want that. But another feature that's not included here is the ability of Armillaria fungi to produce rhizomorphs. And so rhizomorphs are sort of an aggregation of this mycelium into shoestring-like threads. So one of the common names for Armillaria root disease is the shoestring fungus. And those rhizomorphs have the ability to, once the sugar content of this infected tree starts going down and down, the fungus is nibbling it all, it's eating all the carbon in there, it gets to some magic tipping point. And the fungus all of a sudden creates these rhizomorphs, these shoestring-like appendages, and they leave the host, and they start coursing through the soil. And they're looking, go everywhere. They just sweeping underneath the ground through the soil, looking for a susceptible host. Right? And so they have that unique ability to leave that infected host and explore the soil. And so it's left its place of infection, and it can move several meters to find through non-host tissue, grasslands and shrublands.
So, and that's unique to Armillaria. The laminated doesn't do that?
It doesn't do that.
Okay.
Yeah. And another one that you're likely to see in old western hemlock, true fir here, is this heterobasidian root disease. And you can see, here's an old hemlock tree. This is a conch. Here's another conch. And conchs are just woody mushrooms. So toadstools and mushrooms, you think of kind of fleshy, and they wither away within a couple of weeks. These will persist for years on the side of this tree here, and releasing spores day and night, winter, summer, spring, and fall. And so that's a unique mechanism by which this disease is able to spread across the landscape. Anybody growing Christmas trees down this way? True fir Christmas trees, noble firs have real big problems with this particular root disease because any stump that you create is a suitable substrate for these spores to land on. And with Christmas trees, you're creating, 10,000 stumps per acre, a lot of stumps. So it's a real problem in Christmas trees. In older stands, every state park that has western hemlock in it has this particular disease. It's very problematic from a hazard tree perspective because the way the fungus decays, it's not a very fast or very aggressive fungus. So here you can see, here's a woody conch, and the idea is to reflect that out of this conch come spores. These spores move down these cut stumps or any wound on the tree, and they got inside this root here, and you can see all this dark tissue is decay. But what's interesting about this decay is we know that this lighter color here is the sapwood, right? And it's through the sapwood that the water that's being absorbed by the roots moves up this column into the foliage. And the sapwood is not affected at all, right? And similarly, the phloem, or that sugar-rich tissue just beneath the bark, where sugars are moved from the foliage in the canopy down the stem and into the roots, there's nothing affecting the phloem tissue either. So it's a battle, right? Every year that this fungus advances another annual ring of decay, the tree is putting another annual ring of growth. And so over time, you're left with these structures where have a significant amount of decay. And invariably, the primary cause of mortality, the direct cause is wind throw in these particular situations. But, you know, the challenge, for example, in state parks is sometimes they're the biggest trees on the property. They're sort of iconic, right? That's why they were kept there, because somebody decided that, boy, there's no value, economic value in that tree. It's not worth cutting that tree. So it was left, but they didn't appreciate the role that decay was playing. And if you ever put your hand in here, it's pretty soft, and you'd find these white pockets with these little black flecks, which is a sort of a distinctive feature. In the absence of these fruiting structures, it's really hard to tell who's doing the decaying here.
Which fungus is this?
This is heterobasidian.
So it's another one. It's not amaryllis or laminated.
It's not. So the old days we used to call it anosis root and butt rot.
Some of us still do.
Yeah, you're dating yourself.
I
Right.
bet.
So,
Weary-eyed.
so what do we do? Well, we've tried lots of different approaches to control these root diseases. We talk a lot about these days with the DNR. Well, what is the value? We're trying to justify using fire on the landscape. And I was recently asked, can we add root disease control to one of the benefits to promote our prescribed burning program? And I said, well, no, you can't. You know, it turns out that the soil is a very effective insulator. And so just an inch or two below the soil, these fungi are able to live through a fast-moving fire. So although maybe the hosts might be displaced, consumed, anything that's still underground will still be able to support a population of these root disease fungi. In the old days, we did a lot of stumping. There's some significant historic trials over in Glenwood on armillary root disease, where this is really nature's way of cleansing the site of root diseases. These trees we saw earlier. See if I can go back. See what happens here? When these infected trees grow over, the stumps come out of the ground, and it's on these stumps and roots that the fungus is living. Once you get it out of the ground, it gets exposed to the sunlight, and it dries out, and it desiccates the tissue. And it doesn't eliminate it. As you can imagine, there's still, on the underside here, there's still some tissue down there. But what it does, it sort of resets the clock. Areas where, when these trees were still standing, there was a lot of fungus around. Knocking these trees over, we got rid of, 3/4, 4/5 of that. And so, the old analogy of a tree can live, and often does, with well,
Toe.
a wolf chained to your leg. But it can't stand a pack of wolves chained to your leg. That's another story. So stumping was a way to sort of mimic starving these fungi, getting rid of the tissue upon which they live. This is a picture of a fungus that competes with these pathogens, Plebiopsis gigantea. It's, in other nations, it's used as biological control for some of these pathogens. We don't have the appropriate documentation to support releasing this fungus into the soil as a competitor to these pathogens. But that's, biological control is sort of the holy grail of where it is. Just let these fungi duke it out amongst themselves. And, we don't have anything there. So this is an old slide where we use fungicides, methyl bromide and chlorpicrin were used to fumigate these stumps, and they're very effective but have deleterious impacts on the atmosphere. And fertilization was another one that was commonly considered. It turns out that the root chemistry is actually rather complicated, and when we fertilize with nitrogen, we tend to elevate the sugar content in the soil. And that's exactly what these pathogens are looking at. In agriculture, they are able to fertilize with potassium-based fertilizers. Anybody who's familiar with fertilization in the Northwest, since potassium is not a limiting nutrient out here, so it's difficult to get these conifers to take up that potassium. But when used in other systems, the potassium increases the phenol concentration in the roots. And these phenols are defensive compounds, and they're able to, in other systems, to suppress pathogens in the soil. So, just a couple of basics. It's important to know what you have. As we saw earlier, different trees are susceptible to different pathogens. So if you know what you have and where you have it, you may be able to focus your efforts by encouraging tolerant or resistant species. Here's 3 publications on root diseases. They're pretty common. Most people have these. They've been around for a long time. But in each one of them, not surprisingly, there's a table like this. And it lists the different, in this case, conifers and the different diseases. And it has these numbers to represent the relative susceptibility to these various diseases. And a 1, in this case, laminated root rot on Douglas fir, severely damaged, right? And so we have others moderately damaged, seldom damaged, and not damaged. So these are terms that's used in this particular publication. Other publications might use the resistant, tolerant, susceptible, immune, but it's all basically the same idea there. There might be some silvicultural approaches to managing root diseases. We talked about removing stumps and other things. And there's always this do-nothing strategy. And, with small landowners, I see they're very comfortable with that approach. And you can see, here's a pocket of Douglas fir. You can see the large trees at the perimeter that have recently died. Here's an old fallen snag. Here's an old stump. And there's actually a lot of, I walk through these woods. I've been walking through these woods for about 25 years now. And every couple of years you see another tree die. And in this opening are coming up lots of hardwoods. And what's amazing is this is where the birds hang out. This is absolutely where the birds are hanging out in this particular woods. And there's some value. If you haven't heard Ken Bevis's talk on wildlife, most of you sure have. I get a commission every time I stick this slide on Ken. And here's another site, much more advanced site. So this was a mixed stand. Interestingly enough, western hemlock is in the stand, western red cedar, and Douglas fir. This is the last Douglas fir that's standing in this pocket. This is a Douglas fir that's on the ground. This is a pocket. These trees are 400 years old. And this is the last Douglas fir finally taken out by laminated root disease. But not surprisingly, western red cedar is coming up in the understory, and that's likely going to be able to contain the particular pathogen. Interestingly enough, as these trees came down, their root wads popped out of the ground. So, I can see, ecologically, the sequence of disease advancement has been set back almost to time zero. Like the soil is now relatively clean of any pathogenic influence in this area.
We hope.
Do we hope? I mean, it's okay. You don't have a problem with that, do you? Maybe. So that's all I wanted to say about root diseases. Are there any questions about that before we? Yeah.
So how long does it stay in the soil? Obviously, it's not 30 years and 6 months that it's done, but at 50 years, could you replant the Doug fir in an area that was previously infected?
It's largely a function of the size of the stump that was infected. So when I came to the DNR in '96, my predecessor, this was out near Forks area. There was a site that was infested with laminated root disease, and the strategy was we're going to harvest the Douglas fir off this site, and we're going to plant red alder. So by the time I got there, the red alder stand was 30 years old, and the question was, can we go back to planting Douglas fir? So at that time, I walked through the stand, I looked at these stumps, they were still pretty solid. I mean, these were probably 30-inch diameter Douglas fir trees. They were pretty stout. And so my recommendation was no. We could still isolate the pathogen from the soil. So about 2 years ago, I went to revisit this site. And so with a second stand of alder that's now 28 or 29 years or 25 years old, ready to be harvested again. And I don't think that enough time has passed. And so my recommendation was to them, until the day that you can put your boot through that stump. And of course, on a larger stump, it's going to take more time. But I have been to sites where they harvested 12 to 14-inch Douglas fir stumps. And after 25 years, after 20 to 25 years, you can put your boot through that stump. And I say it's safe to go back with Douglas fir. Now, so what do we know about that? Well, we need to avoid stumps, right? If I'm going to plant in these areas, I'm not going to plant next to that paste-hardened stump, right? So I'm going to be spatially cognizant of where those, even though they're old, and even though I was able to put my boot through them, those are hot zones, that I need to avoid. And if they're clustered, maybe I'll plant something else in that area. But you can buy time, right? You can buy a decade by planting it 6 to 8 to 10 feet away from that stump. Because as this tree is growing, this stump is, whatever the opposite of growing is, shrinking. Right. So I guess that's the short, until the day you can put your boot through that stump. It's able to harbor the pathogen, yeah.
Well, let Mark go.
So it was occurred to me about the effectiveness of burning. Can you talk a little bit more about that?
So the strategy with burning was maybe we can get the soil hot enough that it will consume the fungus that's growing on the roots underground, okay? It's relatively easy to burn the stumps out, but it's those roots that are anywhere, at 12 to 24 inches deep, depending on the species. It's unlikely that they will be consumed by the fire. So if your intent is to physically burn them out, unless your fire is so hot that you're likely to do other damage to the soil, it's unlikely that you're going to affect the vitality of those fungi.
So,
What you will do, however, is you will be eliminating these trees, right? So in a sense, you're stopping time for them. And so the fungus now is on notice that it's no longer going to get fresh carbohydrates because the tree, that forest has been consumed. So it's hoping, it's taking up residence in those roots, and it's waiting for a susceptible root system to come its way that it can then infect. And depending on how much time that takes, or which species is grown in proximity, it may or may not be able to do that.
well, on Volga unit, 5 to 10 acres, it's got some root rot. Pile of brush, 10 feet tall, 20 feet wide, don't cut the DNR, it's 20 feet wide, I think it's supposed to go less. Fire, get it going really hot. When we're all done, it's got ashes that deep, and there's no really big fir stumps in it.
Yeah.
I plant some Douglas fir in there. Does that reduce the risk of root rot?
Yes.
Because that's a much hotter fire where that burn pile was
It is.
than a forest fire that sort of just blows through an area.
It
and
does,
It's in the,
yeah.
it's in the crown, we'll say.
Yes, yes. Very different experience for that pathogen. And depending on what proportion of those stumps, you mentioned the pile, what is in that pile? Are you actually yanking out smaller stumps that will easily maneuver into that pile? That's another effect that you're going to be having on the amount of heat that's generated from that. But, the downside is you're kind of limited to that pile. And that's true that there are areas, if you're strategic about it, and if you do preferentially make those piles in the pockets, that would be advantageous to the disease in the area.
Well,
To
what if
controlling
I
those.
plant a fir in the burn pile and then ring the pile with western red seed around the perimeter?
Yeah,
So,
yeah.
just an experiment.
Yeah, we've done the same.
I know you have, it's
Yeah.
in your book. Where do you think I
Oh,
got
oh.
that?
But the fir is still pretty young. I mean, they're 10, 15 years
Oh.
old. They're looking fine.
Yeah.
But
Yeah.
but you don't know until later.
Yeah. Yes.
Can I go back to the alder for a minute?
Yes, you may.
We've not been planting alder for a very long time, but some of us did plant a fair amount of it for a few years. And what we think we saw was that alder does not follow alder very well. When we tried planting alder into a natural alder stand that had been harvested, it didn't do well at all. Now, that was very unscientific, but have you heard anything else or anybody in the DNR looking at that?
I don't have an answer to that.
Okay.
We struggle in the DNR, in our alder plantations, with ice damage
Okay.
and snow damage. And so we're challenged by finding sites that are suitable, that are not at risk of accumulating ice and stem breakage. That's one of our concerns that we have when trying to grow all around the landscape. We don't know where those areas are. And we've bought a lot of other people's alder. And so we struggle with keeping that. Every year, it's, you're crossing your fingers hoping this is not the winter where you get a heavy, wet snow. I don't have any experience on those multiple years planting alder and having problems with that. Anybody else?
This is on armillaria.
Yes.
So we kind of know what to do with laminated root rot. but
Yeah.
I feel like I kind of know what to do after 40 years.
Yeah.
But I don't know what to do. I don't have a species that, what can you follow armillaria with that's resistant?
So here's what I know about, so that, we talked about armillaria being the genus, and Astoy is the species of armillaria that's most pathogenic on conifers. What we know is that within the species, there are an infinite array of clones of Armillaria astoy. And it seems like those clones have a unique identity. In other words, each clone has a preference for what it's best at killing. And so I did a study 30 years ago where I collected a dozen clones of Armillaria. And I had a dozen, maybe 10 conifer species. I also used aspen. And so this very complicated matrix. Each one of these 10 clones was inoculated onto one of these conifer species and all. And what we found was that, wow, there are some clones that were avirulent. Although they belong to the species Armillaria astoy, the most pathogenic species of Armillaria, they were unable to kill aspen, they were unable to kill lodgepole pine, whatever the species was. They had no virulence. They were impotent against these particular conifers. On others, on the other hand, they were very aggressive. Their behavior wasn't uniform across species. And so when I go out into the woods and identify Armillaria as the causal agent, the first thing I want to know is who's the culprit? Like, who's the weak host out here? What is the host that is most susceptible to this clone? Because you're going to have to live with this clone until the day you can put your boot through the stump, which is a little different with Armillaria, because Armillaria is a very effective saprophyte. So it can live on chunks of wood that survived the burn, okay? So when you have a clone of Armillaria, we call root diseases diseases of the site. Like you said, how do I deal with this? Because it's not going away. And so that's what I would do. Is I would look across the landscape and say, well, what is this conifer species in this particular case that Armillaria is preferentially attacking? Because it's likely that it's a single clone. And this particular clone, in order to evolve through time, very few of them have the ability to overcome the defenses of a resinous species like Douglas fir. Oh, that's not working out. I'm going to go over to this western red cedar, not resinous species. A whole other metabolic pathway of chemicals are required to successfully invade that tree. So I don't know. I ask you, who is living? Who is surviving? And that's the one I put my money on.
We'll see. I planted a bunch of cedar in there with a little bit of a hopeful.
Excuse me.
How old are they?
That side about 5 years.
It is really challenging. You know, we talked about these susceptibility rankings. One of the things that throws a wrench is stress and vigor. And so you can have, you know, it's kind of like us. We're here today not because we have no pathogens in our system, right? You know, our immune system is just working. That's why we're here. You know, if I were to take a biopsy of any one of us, I would find an array of nasties in us, right? But that's okay. We get plenty of rest. We get exercise. We're out in the woods. We get a lot of fresh air. We eat healthy. And so we're able to live with these pathogens in our system. But you impose a stress. And that's what we've been experiencing a lot in the last few years. It's not just hot, and it's not just droughty, but it's a hot drought. You know, I've heard that word those two words together more in the last couple of years than I'd ever heard. It's drought. Oh no, we have a hot drought, right? And so that sort of changes the equation for these trees, that all of a sudden their ability to tolerate this onslaught of diseases is really being challenged by their lack of vigor. And so we try to control vigor by, thinning. A lot of stands, there's just too many trees, right? Even on the west side. And so these trees are struggling, and you can see in the structure, the composition of the trees, the size structure, that, God, their trees are really struggling, but we leave them there. We let nature pick and choose for us. But, you're kind of rolling the dice, because she doesn't really care what kind of tree you want. And so sometimes it's the big ones that she takes, and sometimes it's the small ones. You just don't really know. So that's a bit of a challenge, is when our trees are stressed, and how that affects their susceptibility.
Well, maples. I hear you talk, I love your presentations, but I never hear you talk about maples. Ours have been dying for 20 years, and I was accused of starting fires at their base. Which, that's not me starting fires at their base, they just turn black at the base.
Well, I'm sorry, but look who is on my slide here. I'll tell you stories about maples. I got to the agency over 25 years ago, and you've all driven up and down Highway 12, and you've seen the skeletons of bigleaf maples. They're still standing 20 years after they were killed. And I can remember early on going up there, and people would say, my maples are dying, and I'd go up there. I couldn't find anything. Like, I could not find anything for a couple of visits, and then all of a sudden, I said, I'm gonna look more closely, and I started finding armillaria root disease on the bigleaf maples. And I went to this house, and I found armillaria root disease. Okay, the next guy would call me, from another county nearby, and I'd be confident, because, it's and I'd run up there, I'd find armillaria, and it's armillaria. And after about 4 or 5 of those, I wouldn't even go make the visit anymore. I'd say, you live up that way? Yeah, it's armillaria. And so, not surprisingly, somebody called me on it one day and said, I hear you blaming armillaria, but I have one of those Pulaski tools. And I've done some digging, and I can't find any armillaria. I said, I'll be right up there. And we went up, and we practically excavated that tree, and we didn't find any armillaria. And so about that time, I started submitting grants to the U.S. Forest Service. And so the way that the big leaf maple was dying back, I thought it was a vascular wilt. Vascular wilts are very common in these hardwood trees. And so it's a fungus that plugs the plumbing pipes in the stem. And, you can't get water to the ends there, so that's what it looks like when you girdle the tree. The leaves turn yellow and the branches die back. And so I submitted a grant to the Forest Service. We got, $50,000. I sampled hundreds of bigleaf maple trees. I sent these samples to the lab. Not a single sample came back for verticillium, which is the fungus that causes the wilt. Wow. Oh, I had egg all over my face, because I was certain that's what it looked like. So then, people started helping me out. I said, that's all I know. Is it root disease or is it? And so I got in touch with some pathologists at Oregon State University. Oh, no, you got to look in the soil. It's all in the soil, in the fine roots. So we submitted another grant. And we went to all of these trees, to all of these sites, and we dug soil pits. We put the soil in a bag. We sent the bag to the lab, and they were going to look for phytophthora and pythiums and fusariums and other fine root rotters. Armillaria, laminated, heterolithid, and these are all coarse root rotters. You find them at the base of the tree. Whereas these other Pythiums and Phytophthora, they're fine root rotters. You find them way out on the ends, on the tiniest little roots. So we dug soil pits and we sent hundreds and hundreds of samples. Nothing conclusive about the Pythiums and the Phytophthora and these diseases. Interestingly enough, when I look back, all of the samples that we sent were from living trees. They were dying, but they weren't dead. So about a couple of years ago, just 3 years ago, I get a call from somebody up in Seattle Parks. Says, man, our trees are dying, and we don't know what's killing them. And so we go up there, and we isolate this this fungus. You can't really see it on there. So the staining inside, this is a cross section, it's caused by this fungus, Cryptostroma corticale. So this is a fungus that is native to the lake states. There's been one report of this fungus since west of the Mississippi in the last 100 years. And it was in 1968 in Walla Walla, Washington. And so this is a disease that's very common in the lake states. They grow a lot of maples back there, but it hasn't been reported elsewhere. So when we reported it a couple of years ago, oh no, big, threw money at us, try to figure out about this disease. So apparently what happens is this fungus, when the tree is young, like really young, the fungus gets in the tree and it hangs out there for decades. I have no idea what it's doing in there. But then, when the temperature reaches some critical threshold, which we've reached in the last couple of summers, the fungus is no longer happy in the tree, and it grows outward, and it forms these spore mats. This is called, it looks like sooty bark. And so
My fires.
the bark is literally blown off the tree. And out of these this black bark, there's just billions and billions of spores. What's noteworthy about this particular disease is that the spores are a human pathogen, and they cause a disease called pulmonary pneumonitis. Okay, and so this is a disease that you can see reports of it in the New England Journal of Medicine from mill workers in the Midwest. They harvest their maples, they put them in storage, they come back later to process the wood, everybody gets sick because they inhale these spores that infects their lungs. So what turns this otherwise unremarkable fungus? What's interesting, so we did a survey this last summer, and these are two dead trees. It only appears when the tree is dead. But on living trees, so we surveyed every state park in Western Washington. That was our population. We visited every state park in Western Washington and sampled their bigleaf maple. The state park was very generous, but they only allowed us to core into 2 trees at every park. 3. So we did that. And we were only allowed to core into healthy trees. We didn't, they were healthy, so they could tolerate a core. That was fine, but interestingly, virtually in 9 out of 10 of these trees, we cored hundreds of trees. So 185 out of 200 cores, maybe even twice that. These trees that are asymptomatic, they're perfectly healthy. They're iconic, beautiful bigleaf maple trees. They had this fungus in their center. We were able to isolate from the cores. You would never know that. And in every park, we would also find these sooty bark symptoms, where, for whatever reason, like we don't really know, there seems to be a temperature threshold that that we're not sure if it turns on the fungus or it turns off the maple. But there's something going on there. So the bigleaf maple are very sensitive to high temperatures. And those that are growing in marginal sites are the ones that are going to turn out like this. Every park has trees on it like this. See, these are all dead. And we could find the fungus on these dead trees, but we couldn't find the fungus even on these dying trees. Like the trees, it was only in situations like this where you'd find the fungus.
So the graduate student at UW that put some effort into this
Yeah,
a couple years ago,
Benson.
and he came back with climate change.
Yes,
And, I mean, I hear that coming, I hear that that's, can be part of it, but I
Well,
like
well-
what you're saying a lot more.
So I worked on that project with him, and when I started seeing this, I spent a lot of time reflecting on what did I miss, because I went with Mr. Benson to all of these sites, and I can remember very explicitly saying we're not looking at dead trees. We're not looking at dead trees. No, we're not looking at dead trees. We're only looking at living trees. Okay, we're only looking at living trees. So we would have missed it. I mean, I sampled for verticillium, I sampled for phytophthora. I looked at a lot of trees, but every tree that I looked at was alive. And so what he found was that there was an element of disturbance associated with mortality in the study that he did. And then he was able to identify through dendrochronology that there were these trigger years, that there was climactic events, hot droughts, if you will, that occurred over time, that following those episodes, there was mortality. And I suspect that that's what was going on, that this pathogen was in all of those trees. And I suspect that the association with disturbance of those bigleaf maples was the fact that these were trees that were growing on less than optimal sites. And that's why he found the association with the disturbance in his study.